Good morning. It is Dr. DeShavo touching base with you about week one. I thought brief little videos might be helpful. I'm not sure about every week, but maybe every other week as we start to move into new lab material. Once you get the rate and the rhythm of this course, it'll be easy peasy. So here's week one. I've sent the weekly email. This will continue and should be completed by January 21st. You can see that I noted that there in the email. So basically you go through week one online content. Those are my lovely lectures. You'll get to hear my voice over and over again. So I've made those videos in 15-minute chunks. Now, I think it's a great idea. You usually have never more than 6, 7, 8 videos. Do one a day. Memorize it until you're blue in the face and then move on. It does a couple things. It breaks up the material to be a little bit more tolerable. It gives you time to digest it and time to memorize it. How do you memorize it? That's the main secret, right? And if I had that secret, I'd be a millionaire. Memorization is different for everyone. So whatever helps you to memorize the PowerPoints from my lectures as best as you can is what you do. I'll tell you right now, in lecture, all of my lecture questions come from the lecture material and my lecture notes. I'm not one of those tricky people who's going to pull it from somewhere else. In lab, all of your lab questions, there's one exception when we get to spinal tracts, but I will tell you exactly what to do when we get there. All of my lab exam questions come from the lab manual that you will print. So for lecture, study lecture, for lab, study lab. You will memorize your lecture notes and your lab notes to the point where you could come and tell me the answers. That's how well you should know it. Okay. Lecture is multiple choice, true, false, and matching. Lab, you will have to write the answers. So I shouldn't even say this, but for lecture, spelling is not as important as it is in lab. Let me put it that way. Okay. So in lecture, you'll have, like I said, multiple choice, true, false, and matching. I forget how many questions we have on each exam, 50 to 100 questions. I forget the range. They're all different for all the different classes that I teach. And in lab, you will have fill in the answer. There's about 50 answers for every lab exam. Okay, so anyway, getting a little bit ahead of myself. Week one, what you're going to do is go into Engage, which I actually pulled up. You're going to go to Unit 1, which is week 1 and 2. And you're going to come down. I have this important, watch this first. That is actually the video that I sent to you already on how to run the class. Week 1 lesson, you're going to come in. That won't pop up unless you've already been in. Here's your learning objectives. You hit next. You print the notes. You hit next. You play my videos. And you get to hear my lovely voice over and over again. Okay. The other kind of fun thing about these videos, which I forgot about, and these are my basic A&P videos, by the way. I have nervous tissue and nervous system. It is a lot, but these are review. They should be review for you. Now, with these videos, what I did myself, or you can do, I'm not sure you could do it on this one. I apologize. On the future ones, when you go in to play them, once you've reviewed them, you can actually play them at double speed. I usually go through and review the videos before I come into class. So if you want to review them later, you can play double speed and just listen and see how much you can actually picture of what you've memorized. Okay. So then for week 1, you go to the next video, play that. You just work your way right through. Okay. Now your central nervous system. Those are your PowerPoints. Move on to your videos, etc. Okay, so work your way through that. For week 1, there is no quiz. Okay, so you just review the material, take notes. A lot of this should be review for you, and then move on. Here's week 2. Okay, when you go into there, here's your learning objectives. Here's your notes. Print those. Here's the video. I just want to show you how you can do it double speed. So if you do well with listening like I do, you can hit play and then you could double the speed. So suppose you want to review and listen. Turn that off. Okay, you could get through this in 6 minutes. So I'm a very auditory learner. If I were to watch this once, take notes, memorize the notes, and then listen to it again, I would just literally close my eyes and see how much I remember. So just a little tip. From here, if you hit the back button, it'll bring you back to engage. Okay, just work your way through week 2. Okay, so that is week 1, week 2. There is no quiz for week 1. That is a quiz for week 2 right beneath it. Okay, then you move on to lab. So here's lab. What we're doing is histology and overview of the brain. I actually bring the lab up. We'll review it really quickly. We will meet in person this coming Thursday, January 17th. You want to complete as much of lab 1 as you can. I know you can't answer everything. Do the best that you can. 70, 80, 90% of it, you're going to get points. Boom, done. Okay, here's your lab manual. I sent to you as attachment as well. Print that off before you come in. Now, independent assignment. This is due by January 24th. This is part of your grade. This is why we don't meet every week. You're going to have 5 of these. Okay, work your way through that as best as you can. It's a little bit of a difficult one. If you look at the independent assignment, like I said, there's 5 of them. I'm pulling it up right now. I pretty much, with my rubric, look at what I've requested from you and just take points off if you don't have it. If this will ever open. I'll tell you right now, this is the hardest one. So do the best that you can, all right? I explain the sequence of cellular and molecular events that occurs during synaptic transmission. I went over this in my lectures. Look for the synapse, look for the stops. I literally number it and break it down for you, including pre- and post-synaptic events, such as resting membrane potential. Give me the definition of that. And the definition of a refractory period. Literally walk your way through this and give the answers. Please don't make it more than 1 page. Number 2, describe how synaptic transmission dysfunction leads to clinical problems using one of the following neurotransmitters. Pick a neurotransmitter. Tell me how it dysfunctions. You're done. Okay? Give me 3 journal resources. Review articles would be a good starting point. Okay, just literally follow this step by step and you will get your points for this. All right, let me close this out. Now, for lab, I pulled this up. This is your lab book. I'm literally going through and filling out the answers today, and I will have this in the lab at learning stations for each of you as you come in. The general gist for this is a little tough, just so you know. It's histology, which is the study of tissues, specifically looking at nervous tissue. So the general gist of this is we're going to look at a nerve itself, and I actually have a slide in there that's known as a giant multipolar neuron slide, and it is the parts of the neuron and some other structures in the neuron itself that we'll be looking at. Within the lab, I've also given you pictures. So here's a picture, for example, of a neuron, the different parts of it, what's called Nissl body is the inside of it, which you need to define. So once again, if you don't know what a Nissl body is, you look it up. Every time you get to a word, you look it up. Okay, it's easy as that. And then we have some supporting cells you can see. An astrocyte and a oligodendrocyte, for example, are 2 supporting cells, which are known as glial cells. So we'll look at the nerve itself, which is a functional unit of nervous tissue, and some supporting cells that support and nourish the neurons and also provide barriers within the nervous tissue itself. And I'll explain what those are in a little bit. Here's another picture of a neuron right there, the funny looking thing there. Now we talk about dendrites. You should remember dendrites from A&P. It helps to transmit nervous signals away from the cell body of a nerve. Okay, and there's dendritic spines here. So there's a critical thinking question there saying, why do you have multiple junctions off a dendrite to help nervous signals? Okay, what does axodendric contact mean? You probably don't know what that means. You look it up. Okay. Glial cells are going to be cells that support neurons. Okay, you're actually going to have another look at that giant multipolar neuron slide because the supporting cells will be surrounding that neuron. Okay, so we're going to look at these glial cells. They're labeled. You're going to take a peek at that, see if you can tell them apart. It's a bit of a trick question. And then we go into the glial cells themselves. So we actually have bigger examples of glial cells so we can really see what they look like. Here's an astrocyte. In your notes, I had a little brain fart and I put that it helps with the blood-brain barrier. An astrocyte actually does a ton of stuff. It helps to provide functions of neurons, blood supply to different areas of the brain. Okay, somewhat regeneration of neurons. It helps with those. We'll get into all those functions in the future. Oligodendrocyte, we don't have a slide for. An oligodendrocyte helps to create myelin for the axons of the central nervous system. Okay, doesn't help with regeneration of axon processes. That's all in your lecture, so I do review that for you. And that's a picture of an oligodendrocyte, that green and yellow picture. Epidermal cells, I'll have the picture up for that. You could read through what they do. I'm getting a little bit too involved with this. Schwann cell, here's a picture of a Schwann cell. I go over what they do. All these are supporting cells of central nervous system tissue. Dorsal root ganglion is going to be a collection of cell bodies. I will go over this in the future. We'll look at an example of a dorsal root ganglion. Ventral versus dorsal roots of a spinal nerve. I will go over this in week 1 and week 2 lecture and the difference. Okay, these are roots that come off the cord itself and combine to form what's called a mixed spinal nerve. So if we look at this picture, it shows us dorsal nerve roots are here because they're in the back. And ventral are here. Oh, I overshot that. Are here. You know I can't draw. Okay. The reason we can tell the dorsal from ventral, couple of things. Dorsal root ganglion is going to be a collection of cell bodies. Once again, I know you don't know what that is. This is the introduction. You got to look things up and review lecture. And you can see the dorsal roots will feed into that dorsal root ganglion, which is that nice big chunk there. Okay. And then the ventral roots don't have that. And they come down. Can't see it really well, but they will combine to form one mixed spinal nerve. Okay. So dorsal is going to be sensory. I go over this in lecture. Ventral will be motor. And then they'll combine to form a mixed spinal nerve out here. Okay. So that is the transverse view of the spinal cord. So this is just to give you a reference, like we cut the spinal cord right in half, top and bottom, and we're looking down inside that spinal cord. Okay. Pyramidal cell are large cells found in the cerebrum. We're going to go and look at those. They help to provide motor function and motor signals, I should say. Purkinje cells are going to be found in the cerebellum. Okay. And that's another nerve cell in the cerebellum. Now, axons, we're going to look at different types of axons, which are going to be part of the neuron, and they bring information from the cell body away. So dendrites will bring it in, axons send it away. Okay, so we're going to go over some pictures of those, and then we go over an overview of the brain. So directional terminology, what's towards the front, what's towards the back, superior, inferior, you got to look all of these up. Okay, different planes, sagittal, planes are imaginary. Oh, sorry about that. Do I go over planes? I don't do it in here, but that may sound familiar in A and P. Planes are imaginary cuts that imaginarily cut the brain. Is that a word? That cut the brain into right and left, top and bottom. Okay, front and back. We review those. Meninges are the covering of the brain. There are 3 of them. Review them. Know what's on the outside, middle, and inside. They help to provide support and protection for the brain. Okay, cerebral spinal fluid is the fluid that encompasses the brain. It helps to support and somewhat nourish the brain. Okay, ventricles are the spaces where cerebral spinal fluid is created. You need to know where those are. Okay, and general organization of the spinal cord. the levels, cervical versus thoracic versus lumbar. Lobes and fissures are the different regions of the brains, the lobes of the brain, and a basic function of each. And here's some pictures, once again, of the names of the lobes. All right, that is lab 1. I know it's a chunk of change, but we have 2 weeks to do it, and you should have a lot of this done before we come in. You also should be familiar with a lot of it. So in lab 1, we're going to do histology, cells of the supporting cells of the nervous tissue, as well as the neuron itself, the general structures of the brain and the spinal cord, and the inside a little bit of the brain, especially with the ventricles. And that will bring us for the first 2 weeks of class. I will be in lab at 9 a.m. if you need me. And as always, I'm on my email all the time. If you need me, shoot me an email. This is Dr. DeShavo. Welcome to your first unit, weeks 1 and 2, for lecture and lab for neuro, anatomy, and physiology. Have a great day.